Hi everyone, welcome back to Mid Baby YouTube channel. So this video is about gametogenesis, the general embryology in anatomy video. So in this video we'll talk about oogenesis and spermatogenesis in detail, and also various questions asked previously. So we'll focus on the questions. So let's get started. So the gametogenesis, first of all, in order to understand gametogenesis, we just need to know the basics, like what is a pre-embryonic period. And so the pre-embryonic period is from 0 to 2 weeks, and there is embryonic period which is like 3rd till the 8th week, and from the 9th week till the birth of the baby it is called fetal period. So these are various periods: the pre-embryonic period, embryonic period, and the fetal period. So after that, so let's start with the gametogenesis. So first of all, in gametogenesis there is a special cell called primordial germ cell. So this primordial germ cell, it is secreted by the epiblast, or it is derived from the epiblast. And when it is derived, so we get primordial germ cell in the 2nd week from the epiblast. So this primordial germ cell, it is a pluripotent cell. It means that this cell has the ability to form all the germ layers of the body. So in the 2nd week, once the primordial germ cell is formed, it migrates to the endodermal wall of the yolk sac, and the week is like the 4th week of the primordial germ cell migrate to the wall of yolk sac, and by the 5th week the primordial germ cell reach the gonads. So as I said, the primordial germ cell is a pluripotent cell. So, and it means that it can produce all the 3 germ cell layers of the body. So if there is a problem in the migration of the primordial germ cell, usually by the end of 5th week it should be migrated to the gonads. But if there is a problem, like the primordial germ cell is migrating to the neck region instead of the gonad, then we have this craniopharyngeal teratoma. And if the primordial germ cell is migrating to the region between the sacrum and the coccyx, then we have this sacrococcygeal teratoma. So why the teratoma? Because we know that primordial germ cell is a pluripotent, and teratomas are tumors where we have like all the 3 germ layers. So if it is migrating to these regions, then we have this teratomas. So after that understanding, okay, so after that, I would like to mention about various types of potent cells. So we have, first of all, we have this totipotency, and then we have this pluripotency, multipotent cells, oligopotent cells, and unipotent cells. So the totipotency means it is the ability to form the entire embryo as well as the external embryonic tissue. So if we take the example of totipotent cells, the cells up to 8 cell stage in the embryo is totipotent cell. And then comes the pluripotency. So it means the ability to form all the germ layers. So as in case of example, primordial germ cell is a pluripotent cell. Then there is multipotent cell. It means that those cells can produce like more than one category of cells. For example, hematopoietic stem cells are example for multipotent cell. Then there is oligopotency. It means like one category of cell. For example, vascular stem cells are oligopotent cells. Then there is unipotent cells. The unipotent cell can just produce only one type of cell. So the example will be liver cell. It is good to know the various type of potency, and like also if there can be questions like in order to arrange these types of cells from the increasing order of potency, and also it will be like totipotency, then pluripotency, the multiple potent cells, the oligopotency, and unipotent cells. Okay, so before starting into discussing about oogenesis and spermatogenesis, let me talk about some of the basics of cell division. So we know that cell division is of two types. We have the mitosis and the meiosis. So in mitosis, usually it takes place in body cells, and meiosis, we know it takes place in the gonads, the germ cells. So in mitosis, the chromosomal division is equational division, that is 46 chromosome. After mitosis, we get two daughter cells with 46 chromosome each, whereas in meiosis, which is a reduction division, in meiosis the daughter cells will be haploid. It means like half of the chromosome, like 23 number of chromosomes. So, and also the difference between mitosis and meiosis is that in mitosis there is no crossing over taking place. So that means the daughter cells resembles the parent, but whereas in meiosis there is crossing over, and so the daughter cells do not resemble the parent. And also one last thing about meiosis is that meiosis, we know it takes place in like two times, like there is meiosis 1 and meiosis 2, and the steps in meiosis will be like PMAT as a mnemonic. So it will be like prophase, metaphase, anaphase, and telophase. So it is PMAT. So it is prophase, metaphase, anaphase, and telophase. So let me start about oogenesis. So as I said in the starting, like primordial germ cell will reach the ovary in the 5th week. So afterwards, then oogonium from primordial germ cells after cell division forms the oogonium. So by 3rd month, all the primordial germ cell will convert into oogonium, and afterwards the oogonium will make the primary oocyte. So like 5th till 7th month, all the primary oocyte will convert into, all the oogonium will get converted into primary oocyte. And after primary oocyte, there is meiosis 1 taking place. So but in meiosis 1, as I said, there is like prophase, metaphase, anaphase, and telophase, but here it only completes the prophase, and it goes into a resting phase in prophase, which is called diplotting stage, and it remains in the diplotting stage until puberty. So after puberty, only the primary oocyte will complete the meiosis 1. So after the completion of meiosis 1, we have the secondary oocyte and the first polar body. And here in meiosis, this 1, it's not like the division is not equal. So what is first polar body? So polar body are cells without any cytoplasm. So it is why it is formed, because it is formed due to the unequal division of cytoplasm. So the secondary oocyte is getting all the cytoplasm. So that's why we have the secondary oocyte plus the first polar body. And afterwards, the secondary oocyte will undergo meiosis 2. And here in this meiosis 2, it will complete the first two phases, like it completes the prophase and metaphase. Here the secondary oocyte will be in the metaphase arrest, and this arrest will be for 24 hours. This is very important. So meiosis 1, the arrest was, it just complete the prophase, and it was in the resting phase, like diplotting till puberty. And the secondary oocyte completes the meiosis 2. First it completes the prophase and metaphase, and it was in metaphase arrest for 24 hours. And it means that it is waiting, the secondary oocyte is getting arrested for 24 hours because it is waiting for the sperm to come. So it is waiting for the sperm to come. So if there is sperm, so then the secondary oocyte will complete the meiosis 2, and the ovum and second polar body is formed. If the sperm is not coming, then the secondary oocyte, what happens to secondary oocyte is that the corpus luteum will persist, and it will wait for another 14 days and then shrinks and form a white scar, which is called corpus albicans. Albicans means white. So that is all about oogenesis. So if we talk about spermatogenesis, it is not that complicated like oogenesis. In spermatogenesis, like primordial germ cell will reach the testis in the 5th week. So this is the main difference between oogenesis and spermatogenesis. As I said, in oogenesis, then primordial germ cells will undergo oogonium, then there is primary oocyte production. But in case of males, in spermatogenesis, the primordial germ cell is in the dormant phase until puberty. So, and after puberty, only spermatogenesis is actually taking place. So the main difference is that in oogenesis takes place in the intrauterine life itself, but in case of spermatogenesis, it is taking place only after the puberty. So after puberty only something is happening to the primordial germ cell. So after puberty, the primordial germ cells will convert into spermatogonium, and this spermatogonium will undergo like 5 to 7 cycles of division, like spermatogonium dark A, B, and also. So there are some 5 to 7 cycle of divisions, and then after all these divisions, the spermatogonium gets converted into primary spermatocyte. And in this primary spermatocyte, we have this meiosis 1 division. So we have got 2 secondary spermatocyte. So here there is no formation of any polar body. So we have got 2 secondary spermatocyte, and meiosis 2 takes place in the secondary spermatocyte. Then we have, so from 1 secondary spermatocyte we have 2 spermatids. So we have from the secondary spermatocyte, after meiosis 2 we have 2 spermatid, and one special process is happening to the spermatid which is called spermiogenesis. So what is spermiogenesis? It is just a process where rearrangement of organelles is taking place. So basically after the spermatid, the spermiogenesis takes place and converts the spermatid into spermatozoa. So this spermiogenesis is the process of there is a rearrangement of organelles, and also what are the processes take place, like there is shedding of excess cytoplasm, there is formation of the tail and the acrosomal cap of the spermatozoa. So thereby after spermiogenesis, spermatid get converted into spermatozoa. Like we usually get questions like this was a previous questions, like the just write in the order of which is coming first. So we know that spermatogonium, from spermatogonium we get the spermatocyte, then comes the spermatid, then after spermiogenesis we have the spermatozoa. So like from one primary spermatocyte basically we are getting 4 spermatid. So as I said, one spermatogonium, it can like undergo 5 to 7 cycle of divisions. So like from one spermatogonium we can get like 64 to 512 spermatozoa. And some difference of spermatogenesis and oogenesis, as I mentioned, like spermatogenesis only takes place after the puberty, whereas oogenesis it starts in the intrauterine life, and the polar body is formed only in oogenesis, not in spermatogenesis. And also from one primary spermatocyte we get 4 spermatid, whereas in oogenesis from one primary oocyte we get only 1 ovum, rest all of polar bodies formed. Okay. And if we get a question like when spermatogenesis is completed, earlier editions it was said like spermatogenesis is completed in 60 days, then it gets changed into 64 days, but in the latest edition it is like spermatogenesis is completed in 74 days. So the spermatogenesis, if we get a question now, then the exact answer, correct answer will be like 74 days if it is as an option. And the question for answer for most fertile period will be 3 days. So, and if it is asked which 3 days, it will be like the 13th, 14th and 15th day. So it's basically 2 days before the ovulation, 13th and 14th day, and 1 day after ovulation, that is the 15th days. And why it is? Because the 3 days it is said to be most fertile period, because we know that sperm is viable for 48 hours, and the secondary oocyte in oogenesis we saw that metaphase arrest of the secondary oocyte and it waits for 24 hours. So it's like based on permutation combination, it is said to be found to be like 3 days are the most fertile period. Okay, so now the questions comes, as we said, like the fertile period is like 3 days, like 2 days before ovulation and 1 day after ovulation. So now the questions comes when we get to know that ovulation takes place or not. So basically it is through the hormone LH. So if like before the ovulation, 36 hours before, we get LH surge in the body, and like just 12 hours before ovulation there is LH peak, or it can be also said like 12 hours before ovulation, or like at ovulation there will be a LH peak in the body. So usually patients or women is advised to know their ovulation by, like, nowadays LH pads is available. So if it is shown to the arm of the woman, then it shows the alarm, like if there is LH peak or LH surge in the body, and thereby we could know that ovulation has taken place. So lastly, just some one-liner questions, like if in male spermatogenesis, where it takes place in the seminiferous tubules. The maturation of sperms takes place in the epididymis, and the capacitation, it means the conditioning of sperms, and it is happening in the female genital tract, and the capacitation or the conditioning of sperm takes place for 7 hours. So so that's all about this video, the oogenesis and spermatogenesis in detail. I hope the video was useful. Please like, share and subscribe the channel. Thank you.